Hello friends, welcome to Premier UPSC IAS. So today we are going to cover the PT 365 Science and Technology part of the Vision IAS. These are one of the very important series of files released by Vision IAS pertaining to prelims examination. So today we are going to cover the Science and Technology part of the PT 365 for 2023 prelims. Okay, let's start. So, starting with the first topic, biotechnology, genetic modification technology. Okay, what is it? Genetic technology or genetic modification technology is a direct manipulation of DNA of many organisms. The term itself tells you what is the meaning behind it. Okay, including plants, bacteria, animals to alter the desired characteristics. Okay, major approaches to genome editing are one, cluster regulated interspaced short palindromic repeats CRISPR associated protein 9, that is Cas9. So sometimes, not just sometimes, many of the times CRISPR and CRISPR Cas9 have been asked in prelims examination. So this is important topic. We will deal it in detail later on. Okay. But in the genetic modification, there are approaches for genome editing. One approach is CRISPR Cas9, okay, and other is homing endonucleases or meganucleases. This is other. Sometimes these could be asked as an option only, okay, and site directed nuclease SDN. So what are the different approaches of genome editing could be a question, okay. Laws and acts and procedures governing the genome editing in India: Biological Diversity Act, Drug and Cosmetic Act 1947, Seed Act 1968, Protection of Plant Varieties and Farmers' Rights 2009, Food Safety and Standards Act 2006, and Disaster Management Act 2005. This is almost important for mains writing only. Genome editing. Let's go in detail. In a Department of Biotechnology, okay, it is an attached office of Ministry of Science. It is of Science and Technology and is a nodal agency supporting research in life sciences, so research in life sciences and promote large scale use of biotechnology. This department comes under the Department of Ministry of Science and Technology. Okay, what is the mandate? Support research and development and manufacturing and biology, promote university and industry interaction, evolve biosafety guidelines, manufacture and application of cell based vaccines. Okay, announced the first National Biotechnology Development Strategy in 2007, National Biotechnology Development Strategy 2015-2020, and now running it is National Biotechnology Development Strategy of 2021-2025. GM crops always in news every now and then. It is very important topic. Recently, the Department of Biotechnology, that is, which is coming under the Ministry of Science and Technology, issued guidelines for safety assessment of genome edited plants. Okay, so those plants which were edited by genome editing, how to have the assessment? Those guidelines were given by Department of Biotechnology, easing norms for research into the genetically modified crops. Guidelines exempts genome edited plants falling under the categories of SDN1 and SDN2. Okay, so all those genome editing categories, but except SDN1 and SDN2, guidelines are given. For manufacture, use, imported, export, and storage hazardous microorganisms, genetically engineered organisms or cells. Rules 1989 under the Environment Protection Act 1986. So these were all the concerned topics when it comes to guidelines given under the GM crops by Department of Biotechnology, but except those under the categories of site directed nuclease, okay, that is SDN1 and SDN2. It is based on an early exemption by the Ministry of Environment, Forest and Climate Change to avoid a long process for approval of the genetically modified crops. Okay, guidelines for safety assessment of genome edited plants 2022. Aim provides regularity, framework, and scientific guidance on data requirement in context of research and development of genome edited plants. Scope of the guidelines limited to plants and products therefore developed using genome editing techniques employing site-directed nucleases. Guidelines notified by Ministry of Environment, Forest and Climate Change under the Environment Protection Act 1986. Okay, the guidelines were notified by MOFC under the Act actually 1986. Okay, Exemption: researchers who use gene editing technology to modify plant genome from seeking approvals from Genetic Engineering Appraisal Committee. Okay, all requirements to develop transgenic seeds will apply to gene edited seeds except clauses requiring GEAC permission. Significance of guidelines: they accelerate the genetic improvement of crops. Okay, the guidelines cover manufacture, use, import, export, and storage of hazardous microorganisms, genetically engineered organisms, or cells rules 1989. Okay, all those are covered under this one. Okay, do you know that BT cotton is a genetically modified crop? In this, a gene from a soil bacterium called Bacillus thuringiensis is used to protect a plant from pest attack. So it is static part, but one of the important point you should know when it comes to BT cotton, it is only GM crop approved for commercial cultivation in 2002 in India. So there are many GM crops. One of the GM crop is BT cotton, and BT cotton is the only GM crop that is commercially approved. Okay, advantages of the GM crops. Okay, increase productivity and prevent the crop loss, food security, enhance nutritional quality, maintain the soil fertility. So maintain soil fertility will be maintained by GM crops. Concerns are environmental concerns. For example, genetic contamination creating super weeds could be possible. Okay, economic false claims about the field, that could be a problem with the economy part of the GM crops. Biosafety of the human and animal health. Ethical excessive corporate dominance could happen when it comes to GM crops, so that could be concerns. Moving forward, first of all, let's see the static part of GM crop. Um, Genetic modification in plants. Cells are grown in tissue culture, which develop into plants, and seeds produced by these plants will inherit the new DNA. Indian Council of Agricultural Research (ICAR) promotes the science-based innovative technology, including research on GM crops. Network project on functional genomics and genetic modification crops was launched by ICAR in 2005. Genome editing technology and transgenic technology both can alter the genome of an organism. So genome editing, which we have seen earlier, and also transgenic technology, both can alter the genome of an organism. However, there is a difference. Okay, in genome editing is a manipulation of genome of organism itself. Okay, but in transgenic technologies, genome of an organism is altered by introduction of one or more foreign DNA, okay, sequences from another species by artificial means. That is the difference. Various bodies on GM crop regulation. Recombinant DNA Advisory Committee (RDAC) monitors the developments in biotechnology at national and international levels. It functions under the Department of Biotechnology. Institutional Biosafety Committee approves low-risk experiments and ensures adherence to prescribed safety guidelines. It also functions under the Department of Biotechnology. Review Committee on Genetic Manipulation. Okay, this also functions under the Department of Biotechnology. Department of Biotechnology come under the Ministry of Science and Technology. Okay, State Biotechnology Coordination Committee acts as a state-level nodal agency to assess the damage due to release of GMOs and to take on-site control measures. Okay, so in the state also there is a coordination committee. District level committees are also present, act as a nodal agency at district level to reports to SBCC or the GAC about compliance or non-compliance of the regulatory guidelines. Genetic Engineering Appraisal Committee (GEAC) is very important. The headquarters in New Delhi, India. It comes under MOFC. Department of Biotechnology comes under Ministry of Science and Technology, whereas Genetic Engineering Appraisal Committee comes under Ministry of Environment, Forest and Climate Change, that is MOFC. Responsible. Large scale use of hazardous microorganisms and recombinants, release of genetically engineered organisms and products into environment, taking punitive action under the Environment Protection Act 1986. Okay, so punitive action can be taken by GEAC. Presently, it has 24 members and it meets every month. Chaired by Special Secretary or Additional Secretary of the MOFC. Important point, static question could be asked. GAAC, that is Genetic Engineering Appraisal Committee has recommended the environmental release of transgenic hybrid mustard Dhara mustard hybrid 11, that is DMH11. Dhara mustard hybrid 11, DMH11. So it has recommended the environmental release of transgenic hybrid mustard. Okay, commercial use under Indian Council of Agriculture Research Supervision and will be subject to Seed Act 1966. About the Dhara mustard hybrid level, DMH level. This could be direct a question in films, DMH level. Okay, developed by Delhi University Center for Genetic Manipulation of Crop Plants. Developed by DMH11. It was developed by Delhi University Center for Genetic Manipulation of Crop Plants. Process used crossing Indian mustard variety Varuna Barnais line with an East European early Hera 2 mutant Barster. So sometimes, these two terms could be itself a question. These terms are seen pertaining to which topic could be a question and options will be given. So the process used was crossing between the Indian mustard variety Varuna Barnes line with an East European early Hera 2 mutant Barster. Okay, contains two alien genes isolated from a soil bacterium called Bacillus amyloliquefaciens. Okay, first gene, that is Barnes, codes for a protein that impairs pollen production and renders the plant into incorporated male sterile. Second, it is the Baster gene that blocks the action of Barnes gene. Resultant progeny is both high yielding and capable of producing seed or grain. Let's see the new term. Bioeconomy is an economy where the basic building blocks for materials, chemicals, and energy are derived from renewable biological sources. That's it. Okay. Moving forward, the Haryana government issued the NOC, no object certification, to conduct the field trials on BG2RRF, herbicide tolerant and insect resistant variety of BT cotton. Okay. So far, India has allowed commercial use of BG1 and BG2 GM cotton, while BG2 RRF approval is pending at various stages. BG2 RRF can provide protection against the pest attacks such as the American bollworm. So, this could be a direct question sometimes. Moving forward, Site-Directed Nuclease Technology (SDN) technology. Department of Biotechnology revised the guidelines for plants and products thereof developed using genome editing techniques employing site-directed nucleases. So let's see what is site-directed nucleases (SDN). SDN refers to the practice of cleaving DNA strands to affect the subsequent genome editing. SDN technology takes advantage of targeted DNA break and hosts the natural repair mechanisms to introduce specific small changes at the site of DNA break. Main SDN technologies are zinc finger nucleases (ZFNs) and transcription activator like effector nucleases (TALENs). So, ZFNs and TALENs are nothing but SDN technologies. SDN standing for site-directed nucleases. There are three types of SDN: SDN1, SDN2, and SDN3. SDN1 produces a double-stranded break in the genome of a plant without addition of a foreign DNA. Okay, without addition. SDN2 produces a double-stranded break, and while the break is repaired by the cell, a small nucleotide template is supplied that is complementary to the area um, of the break, which in turn is used by the cell to repair the break. So this small complementary nucleotide is added it in SDN2. SDN3 also includes a double-stranded break in the DNA, but is accompanied by a template containing a gene or other sequence of genetic material. But in SDN2, it is nucleotide, but in SDN3, the addition is gene or other sequence. Okay, the cell's natural repair process then utilizes this template to repair the break, resulting in introduction of the genetic material. So that's all about the SDN and the SDN applications or types. Depending on the nature of the edit, the process divided into three categories: SDN1, SDN2, and SDN3. That just we have seen. SDN1 and SDN2 do not produce new plant varieties that fall under the scope of GMO legislation. This is very important. Okay, they does not produce new plant varieties. SDN1 and SDN2. In SDN3, the newly developed in SDN3, The newly developed plant falls under GMO legislation only if foreign DNA exceeding 20 base pairs is inserted. CRISPR-Cas9. Let's see this one, okay? It's been 10 years since the microbiologist Emmanuel Charpentier and biochemist Jennifer Doudna published research that paved the way for CRISPR-Cas9 gene editing. Both were awarded the Nobel Prize for the same in 2022. About the clustered, regularly interspaced short palindromic repeats Cas9. Let's see what is it, okay? CRISPR is a genetic code or a sequence found in a bacteria. This is fact-based question could be asked from here. Formed due to the activity of previous bacteriophages that had infected them. Bacteria uses CRISPR sequence to remember each specific virus that attacks them by incorporating virus DNA into their own bacterial genome. So the formation of CRISPR is actually a inherent character of bacteria. So if a virus attacks the bacteria, then bacteria remembers that virus by this CRISPR sequence. This gives the bacteria protection when a specific virus tries to attack again. CRISPR-associated protein 9, that is Cas9, a bacterial RNA-guided endonuclease. So Cas9 is a RNA-guided endonuclease in the bacteria that once activated will make special enzymes that seem to have evolved with the CRISPR. So that's about the CRISPR and Cas9. How does the CRISPR work? There are three key elements of the CRISPR Cas9 work. Guide the RNA, a piece of RNA that locates the targeted gene. This is engineered in a lab. Cas9, the scissors that snip the undesired DNA out. DNA, the desired piece of DNA that is inserted after the break. So that is how it works. Challenges are ethical challenges, concerns over designer babies, ecological impacts, gene with negative traits throughout a population. So that's all about the CRISPR-9 Cas9. Okay, let's see. Sometimes terms itself could be very important. Question could be asked from here. DNA, RNA. DNA, deoxyribonucleic acid, is a hereditary material in most of the living organisms. RNA or ribonucleic acid molecules are single-stranded nucleic acids. Okay, they are hereditary material in some viruses, RNA viruses. Okay, genes. A gene is a specific section of DNA. It is involved in making the RNA transcription or protein production, that is translation. Genome. A genome is an organism's complete set of DNA. Okay, it includes all chromosomes. It includes all chromosomes which house the DNA and genes. Molecular scissors act as a scissors that cut out the unwanted DNA. They are also called as restriction enzymes. Okay, because of their ability to restrict the unwanted genes at the site identified by the guided DNA. Okay, working of CRISPR-Cas9 process. Because this CRISPR-Cas9 process is been under discussion for a long period of time, sometimes the procedure could be asked as steps. Okay, you have to just remember these steps. For example, step one, take as the patient Susan. Susan, if having a sickle cell disease caused by a single DNA letter mutation, then that Susan's DNA, double DNA helix with sickle cell mutation is taken and guide RNA is attached to it. Then in the lab, scientists create a strand of guide RNA that matches the mutated DNA sequence. Okay. Then the third steps comes attaching of Cas9. Cas9, also called as molecular scissors, is added to the RNA mix which was produced in the lab. Then in the step 4, Susan receives an injection of RNA mix Cas9 mix. Okay. Then in the fifth step, guide RNA identifies the mutated DNA that causes sickle cell disease. Then at sixth step, Cas9 cuts on the affected sequence. Okay. Now healthy DNA strand is present in the Susan. Then comes scientists insert a healthy DNA sequence to replace the clipped sequence. So if all goes smoothly, Susan no longer suffers from the sickle cell disease. So that's how CRISPR-9 can be used for benefit in diseases. Advancement in India. Study for treating sickle cell anemia. Research at National Agri-Food Biotechnology edited the banana genome to improve its nutritional quality and pathogenic resistance, etc. Benefits of CRISPR. Faster and more efficient diagnosis of rare genetic diseases. New developments in pharmaceutical and biotechnology. Food security. Sometimes question could ask like in what ways CRISPR-Cas9 can be used, etc. Almost all the options will be correct, but you have to just check also. chimeric antigen receptor T, CAR T cell therapy. This is also one of the commonly seen therapy in the news. Okay, this is the first time that CAR T therapy indigenously developed by IIT Bombay and Tata Memorial Center was tested on patients in India. Research is funded under the National Bioform Mission by Biotechnology Industry Research Assistant Council BIRAG. BIRAG is a public sector enterprise set up by the Department of Biotechnology. Okay, let's see what is CAR T therapy. It is a way to get immune cells called as T cells to fight cancer by editing them in the lab. T cells are a type of white blood cells that attack foreign pathogens. T cells are taken from the patient's blood and are edited by adding a gene for a man-made receptor called CAR. CAR is a special receptor created in laboratory that is designed to bind to certain proteins on cancer cell. CAR is then added to T cells. This helps them better identify specific cancer cell antigens. These changed T cells called CAR T cells. CAR T cells are then given back to the patient. Benefits of the CAR T therapy. While existing treatments work towards increasing life of patients, CAR-T technology holds promise of curing certain types of cancers. Unlike chemotherapy, CAR-T is administered only once to a patient. Short treatment time needed and more rapid recovery through the CAR-T. Okay. For this, let's see. It's almost easy to understand. Okay. Antigen. Any substance that causes your immune system to produce antibodies is called as antigens. An antigen may be a substance from the environment such as chemicals, bacteria, virus, or pollen. Any antigen may also form inside the body. Okay. Different types of white blood cells, that is just a static part. T and B cells, okay? Immune system cells, innate and acquired. Innate immune cells, body's first line of defense that quickly respond to fight infection. Acquired immunity, also called as adaptive immunity, uses T cells and B cells when invading organisms slip through first line of defense. T cells and B cells evolve from learned experiences so take longer to develop, tend to live longer than innate cells. B cells and T cells are also called as lymphocytes. B cells produces antibodies that are used to attack invading bacteria, viruses, and toxins. These antibodies are Y-shaped proteins that are specific to each pathogen. Specific to each pathogen. So this is B cells. T cells are direct fighters of foreign invaders and also produces cytokines, which are biological substances that help activate other parts of the immune system. T cells destroy body's own cells also that have been taken over by viruses or become cancerous. Okay. T cell B cell questions are directly expected in prelims. Genome sequencing. In a recently published research, an international team described first ever sequencing of a complete human genome. Okay. Let's see about genome sequencing. Genome sequencing means deciphering the order of DNA nucleotides or bases in a genome in order of adenine, cytosine, guanine, and thymine that make up an organism's DNA. A genome is an organism's complete set of DNA, include all chromosomes which houses DNA and genes. Human genome contain about 3 billion base pairs. Okay. Applications of genome sequencing. Greater understanding of evolution and biology, identifying genomic causes of rare diseases, neurodegenerative conditions, cancer, etc., identifying genetic mutations in viruses, its evolution and spread. Moving forward, mm, Indigen program aims to undertake whole genome sequencings of 1,000 Indian individuals. So, Indigen, representing diverse ethnic groups from India, funded by Council of Scientific and Industrial Research (CSIR). Genome India project aims to collect 10,000 genetic samples. Okay, genome sequencing of 1,000, 10,000 genetic samples. Okay, across India to build a reference genome. That is Genome India project. It is by Department of Biotechnology. This is funded by Indigen, funded by CSIR. Human Genome Project. International research effort to determine the DNA sequence of the entire human genome. It began in 1990 and completed in 2003. It was coordinated by National Institute of Health, USA, and Department of Energy, USA. Genomic surveillance, okay, with outbreaks of several pathogens and their variants, including COVID-19, monkeypox, etc. In recent years, the need to build a sustainable system for genomic surveillance is felt. Okay, genomics. Genomics refers to study of the entire genome of the organisms and the large amounts of corresponding data generated from it. Genetics, on the other hand, is concerned with the study of heredity only. Genomic surveillance. Let's see what is genomic surveillance. Okay, in genomic surveillance, it involves constantly monitoring of pathogens, okay, and analyze the similarities and differences of the pathogen. It helps to find the monitor diseases, okay, control different pathogens. This is very important. Tailor the interventions and recommendations for the public, how we can intervene, develop countermeasures like vaccines, etc., stamp out the diseases to completely eliminate the diseases. These are the help of genomic surveillance. Okay, applications of genomics: human genomics for medical purposes, microbial genomics for medical purposes, agricultural and aquaculture genomics, biological and medical research, and others like forensic science, assessment of ancestry, etc. Genomic surveillance in India. Genome Regional Genome Sequencing Laboratories (RGSL) serve as the regional hub for genome sequencing. Indian SARS-CoV-2 Genomics Consortium (INSACOG) was established to expand whole genome sequencing of the SARS-CoV-2 across the nation. Okay, Indian Tuberculosis Genome Surveillance Consortium is proposed on the lines of INSACOG. It is proposed. These are established. INSACOG, jointly initiated by Ministry of Health and Family Welfare, Department of Biotechnology, with Council for Scientific and Industrial Research. So, Ministry of Health and Family Welfare, GBT, and CSIR, and Indian Council of Medical Research. All these organizations jointly initiated the INSACOG, a consortium of over 50 laboratories. WHO's Science Council issued its first report on accelerating access to genomics for global health. The Science Council of WHO, established 2021 by the Director General of WHO to provide the guidance on the science and research strategy of the organization. Cloning. Recently, China has successfully cloned a wild Arctic wolf for the first time in the world. Let's see what is clone. Copied material which has the same genetic makeup as the original is referred to as clone. Process of producing genetically identical copies of biological entity, that is genes, cells, tissues, and even entire organisms, either by natural or artificial means. So cloning can be done in both natural means and artificial means. Okay, not identical. Despite having the same genetic material, clones do not always look identical, as environment also play a role in deciding the physical feature of the organism. This is very important. In mammals, including humans, identical twins, monozygotic twins, are natural clones. These twins are produced when a fertilized egg splits, creating two or more embryos that carry almost identical genetic material. Applications of the cloning: help in studying the diseases, recreating extinct species, more milk or meat in the livestock by cloning. Okay, and cloned embryos could produce the stem cells. Three different types of artificial cloning: gene or DNA cloning, transfer of a DNA fragment from one organism to a self-replicating genetic element such as a bacterial plasmid, that is gene or DNA cloning. Reproductive cloning. Okay. Reproductive cloning, transferring nuclear material isolated from a somatic cell into a enucleated oocyte, that is egg cell. Then this oocyte is stimulated to divide, forming an embryo with same genome as the nuclear donor. This process is called as somatic cell nuclear transfer, SCNT, used in reproductive type of cloning. This process was used to create dolly sheep, the first mammal clone. Okay, therapeutic cloning is like reproductive cloning till the production of the embryo. The produced embryo is then grown in the laboratory, that is called as therapeutic lab cloning. Indian Biological Data Center. Indian Biological Data Center was inaugurated at Faridabad, Hyderabad. No, Haryana, sorry. First national repository for life science data in India. Also, as per the Biotech Pride guidelines released last year, IBDC, that is Indian Biological Data Center, is mandated to archive all life science data generated from the publicly funded research institutes. Okay. Objectives of the Indian Biological Data Center are to develop web-based tools for data sharing, perform quality control, curation of data, and data backup, etc. Provide IT platforms for archiving the biological data, development of SOPs for storing and sharing data as per FAIR principles, organize training programs on big data analysis and data sharing benefits. It was established at Regional Center for Biotechnology, Faridabad, in collaboration with National Informatics Center. Okay, digitalized data will be stored on a 4 petabyte supercomputer, that is Brahma, supported by Department of Biotechnology (DBT). Data sharing as per FAIR, that is findable, accessible, interoperable, and reusable principle. Fair data principles were proposed by a consortium of scientists and organizations to support the reusability of digital assets. Also contains the genome sequenced by the INSACOG. Biotech Prize, that is Biotech Promotion of Research and Innovation through Data Exchange Guidelines 2021, released by the Department of Biotechnology. Okay. Under the IBDC, currently two sections have been developed: Indian Nucleotide Data Archive (INDA) and Indian Nucleotide Data Archive Controlled Access, okay, INDA CA. Data access types of IBDC are open access or time release access and restricted access. Biological data means all information related to living organisms, including their nucleic acids, protein sequence, metabolites, and other molecular and functional characteristics. Okay. Maharashtra Cabinet approved the Gene Bank project. It is a type of biorepository to preserve plants by collecting seeds, plant tissue, cultures, and animals by collecting sperms and eggs. At national level, several such facilities are established, such as National Gene Bank at National Bureau of Plant Genetic Resources (PUSA). It is world's largest refurbished gene bank. National Animal Gene Bank at the National Bureau of Animal Genetics Resources, Karnal, and Indian Seed Vault at Changla, Ladakh. Sometimes which bank is located at which site should be a direct question. Mm. Biological Research Regulatory Approval Portal, BioRRAP. It was developed by Department of Biotechnology to track the regulatory approvals for a research proposal on a single portal. Okay, molecular motor. Scientists have built a molecular scale motor using the DNA origami method. Okay, molecular scale motor using the DNA origami method, involving folding of DNA to create a 2D, 3D objects at nanoscale. Molecular motors are a class of proteins that drive intracellular movements. So, molecular motors are proteins that drive the intracellular movement by converting chemical energy into mechanical energy. Some of the examples are molecular motors' role in our bodies are muscle contraction, mitosis, etc. Second topic, IT and computer. Geospatial technology. Geospatial data. Geospatial data is information that describes objects, events, or other features with a location on or near the surface of Earth. It includes vectors and attributes, that is, points, lines, polygons, and other descriptive information about a location. And raster and satellite imagery get a bird's eye view of what Earth looks like via a high resolution imagery. Users of geospatial technology in the GPS or spatial analytics, desktop, web or cloud, and in mobile, in the GNSS and positioning, navigation, surveying, indoor positioning, Earth observation of aerial mapping, UAVs, drone, satellite, remote sensing, etc. In scanning like LiDAR, laser scanning, and radar. National Geospatial Policy. The Ministry of Science and Technology has notified a citizen-centric national geospatial policy in 2022. The vision and goals of such a policy are to make India a world leader in global geospatial space, and integrated policy to move towards a digital economy and improve service to the citizens, better utilization of data, encouraging participation of the private sector. Okay, strategies and approach of these national geospatial policy are Atmanirbhar Bharat via locally available and locally relevant maps and geospatial data, integrated geospatial information framework IGIF, data and information and communications technology infrastructure, innovation, startup initiatives in the geospatial sector, open standards, open data and platforms. Draft National Data Governance Framework Policy. Okay, the Ministry of Electronics and Information Technology released a revised draft of the National Data Governance Framework Policy, National Data Governance Framework Policy (NDGFP). About the NDGFP is it is the revised version of Draft India Data Accessibility and Use Policy 2022. Okay, it aims to enhance the access, quality, and use of data in line with the current and emerging technology needs, objectives to accelerate the digital governance, have standardized data management and security standards. Okay, maximizing the data-led governance and modernize the government's data collection, promoting digital economy and enabling for creating data privacy standards. Okay, types of non-personal data are public non-personal data, community non-personal data, and private non-personal data. Applicability. All the government departments and entities, all non-personal data sets and data and platform rules standards governing its access and use by researchers and startups. Components under the NDGFP are Indian Datasets Platform, Indian Data Management Office, Indian Data Management Office, and Data Management units. IDMO functions in Indian Data Management Office functions in three ways. Creation of the Indian Datasets platform, safeguarding the data usage, okay, safeguarding the data usage and capacity building of the government bodies. Governance structure of National Data and Analytics Platform. Niti Aayog launched the National Data and Analytics Platform (NDAP) for open public use, a user-friendly web platform accessible to all users. Okay, governance structure of National Data and Analytics Platform are high-powered interministerial steering committee, technical advisory group, project management unit, technology vendor to provide direction and oversees progress, to provide expert consultation to coordinate with different stakeholders and for development of the operation of NDAP. Bank for International Settlements, BIS, okay, has endorsed the India's Data Empowerment Protection Architecture, DEPA. DEPA is a joint public-private effort for an improved data governance approach. DEPA forms the final layer of India stack to utilize a unique digital infrastructure, allow users to share their data on their own terms through a third-party entity, consent managers. DEPA's first application has been in the financial sector. It is being tested in the health sector as well. YOTA D1. It is a YOTA D1. It is country's biggest and North India's it is country's biggest and North India's first hyperscale data center that was recently inaugurated in the Greater Noida, Uttar Pradesh. About the data center, dedicated secure space within a centralized location where computing and networking equipment is concentrated for collecting, storing, and processing large amounts of data. Okay, fifth generation 5G. The 5G testbed has been developed as a multi-institute collaborative project by 8 institutes led by IIT Madras. It is the country's first 5G testbed to enable the startups and industry players to test and validate their products locally. Let's see about the 5G technologies. Okay, the different types of technologies here: 3G versus 4G versus 5G versus 6G. Okay, 3G deployed during 2004 and 6, bandwidth was 2 Mbps, latency was 100 to 150 milliseconds, speed average was 144 kbps. 4G launched during 2006 and 10, bandwidth 200 Mbps, latency was 20 to 30 milliseconds, 25 Mbps was the average speed. 5G deployment 2020, more than 1 Gbps, less than 10 milliseconds latency, and 20 to 200 to 400 Mbps is the average speed. 6G expected to launch in 2028-2030, 1 Tbps less than 1 microsecond, about 50 times faster than the 5G will be the 6G. 5G is the fifth generation of cellular networks. Okay, 5G spectrum. Let's see the features. Low band, lower than 1 gigahertz on the spectrum chart. Wider coverage, lower than 1 gigahertz on the spectrum chart. Wider coverage but limited improvement in speed and latency for thinly populated areas and provides indoor coverage in inbuilt areas. 1 gigahertz to 6 gigahertz range, 24 gigahertz band and higher. So these are the features of 5G. 5G is an amalgamation of various technologies such as massive multi-user MIMO, multiple input, multiple output enabled network, small cell stations, mobile edge computing, beam forcing, a laser beam between transmitting entity and the user. 5G operates at a higher frequencies and is designed to connect virtually everyone and everything together, including machines, objects, and devices. Have an enhanced throughput to handle more simultaneous connections. Let's see new terms. Network slicing. Okay. Network slicing is a network configuration that allows the multiple networks. It is an essential component of all the 5G architectural landscape. Each slice or portion of the network can be allocated based on the specific needs, use case, or customer. Fiberization. Let's see the term. Other steps taken by the government to boost 5G are Cellular Operators Association of India (COII) has formed the 5G India Forum 5GIF. National Digital Communication Policy 2018 lays out the objectives with respect to 5G service in India. Fiberization. Process of connecting radio towers with each other via optical fiber cables is called as fiberization. Connects core of the network to edge, facilitating large amounts of data used in 5G services. Provides additional bandwidth and stronger backup backhaul support. In India, currently only 33% of the towers are fiberized, compared to 80 to 90% in US, Japan, and China. 5G Vertical Engagement and Partnership Program VEPP initiative. Department of Telecommunications DOT has invited expression of interest for initiative to enable close collaboration between user verticals and 5G tech stakeholders. 5G Airwave Interference. Director of General of Directorate General of Civil Aviation DGCA flagged concerns over likely interference of the 5G C-band spectrum with aircraft radio altimeters as both operate in mid-C band frequency range. Okay. For telecom service providers, C-band ensures coverage as well as high bandwidth, resulting in faster internet speeds for rolling out of 5G services. A radio altimeter provides direct height above terrain information to various aircraft systems, and use of altimeters in C-band ensures highly precise measurements. Okay. Private captive 5G networks. Union Cabinet has allowed private captive 5G telecom network in India. A private captive 5G network is a network set up by a private entity for the use of enterprise concern and no one else. 5G spectrum auction. A record over rupees 1.5 lakh crore worth of 5G telecom spectrum was sold in recently held auction. Okay, E-band. Broadband and Wi-Fi players opposing giving E-band exclusively to telcos. E-band is the range of radio frequencies from 60 GHz to 90 GHz in the electromagnetic spectrum. E-band is a major solution to 5G's microwave transmission. 5G Open Radio Access Network (RAN). Center for Development of Telematics (CDOT), VSIC Networks Private Limited and VVDN Technologies Private Limited have signed an agreement for developing 5G Open RAN and other products together. RAN is a part of network that connects the main network infrastructure to end users. It connects the individual in devices to other parts of a network through radio connections. It provides critical technology to connect users to the mobile network or over radio waves. Benefits of Open RAN: data center economics to radio network, vendor diversity, agility of innovation, faster time to market, automation and manageability, lower total cost of ownership, and intelligent and programmable networks. Idea of Open RAN is to enable operators to mix and match the components. Open RAN will create a multi-supplier RAN solution. Standalone 5G, that is SA5G, a telecom network where both core network and radio access network RAN are upgraded to 5G. It replaces the LTE network 4G wireless communication standard and allows for completely independent operation of a 5G service without any interaction with an existing 4G core. Non-standalone 5G, in this only RAN part of the network is upgraded. It is built over an existing 4G network. Advantages of standalone over non-standalone are superior voice quality, significantly more speed, lower latency, etc. Challenges of the standalone 5G are not all phones will have support of the standalone 5G, will cost more, etc. Satellite communication. Recently, TRI releases recommendations on licensing framework for establishing operating for Satellite Earth Station Gateway (SESG). SESG, also known as Gateway Hub, acts as a bridge between space-based communication network and terrestrial communication network. It houses the equipment that convert the radio frequency signal into an internet protocol signal for terrestrial connectivity. Satellite communication. It refers to any communication link that involves the use of an artificial satellite in its propagation path. Example: GSAT-10 communication satellite, INSAT-3A, etc. in India. SATCOM has two components. Ground segment consists of fixed or mobile transmission reception equipment. Space segment, that satellite itself. Advantages of satellite communication: cheaper for long distances, can connect remote locations, expand reach of telecom operators, used in global communication. Satellite internet services is a wireless internet connection which uses satellites in space to get an internet signal from internet service provider to users. Okay, satellite based connectivity is possible using geostationary, medium and low earth orbit satellites. It is different from satellite television as internet signals go both ways with a much greater amount of bandwidth to move all that data. Broadband signal is sent and received to satellite in space rather than using an underground copper or the fiber network. Wi-Fi. RailTel, a mini Ratna PSU under Ministry of Railways, has launched the PM Vani scheme based access to its public Wi-Fi services across 100 railway stations. Wi-Fi network can also be accessed through mobile app Y-Dot built by RailTel and C-Dot. Splinternet. Russia-Ukraine war has threatened to splinter the internet. Splinternet refers to internet that is increasingly fragmented due to nations filtering content or blocking it entirely for political purposes. For example, China's Great Firewall keeps American tech giants out while pushing online services developed indigenously. Russia in 2019 passed sovereign internet law, or the online Iron Curtain, that enable the country to disconnect its internet from the rest of the world. Gigamesh. Gigamesh is an innovative wireless network solution that can give fiber-like bandwidth internet services to suburban and rural areas. It aims to address the congestion issues in 4G infrastructure developed by Astrom. Astrom is supported by Technology Innovation Hub at the Indian Institute of Science. This is about Gigamesh. Let's see what is VPN. Virtual Private Network. VPN is an encrypted connection over the internet from a device to a network in real time. Okay. VPN, use device, VPN client, internet service provided, encrypted, here encrypted, VPN server, and here it is internet. This is also connected. But if there is no VPN, then use device, direct in internet service provided, there is no VPN client, and direct to the internet, there is no VPN server. Okay. Recently, the central government has asked virtual private network companies to keep a record of their logs of customer information as directed by the Indian Computer Emergency Response Team CERT-IN. Okay. Indian Computer Emergency Response Team. About CERT-IN guidelines. New directives by the CERT-IN under Information Technology Act 2000 state that all cloud services and the VPN providers to maintain a series of extensive customer information for at least 5 years, even after the cancellation or withdrawal of the registration. Data center companies and cryptocurrency exchanges are also asked to collect and store the user data. Indian Computer Agent Emergency Response Team (CERT-IN) is a functional organization of Ministry of Electronics and Information Technology. Types of VPN: Secure Sockets Layer (SSL) VPN, Site-to-Site VPN, and Client-to-Server VPN. The Secure Sockets Layer (SSL) VPN, it enables individual users to access an organization's network and client-server applications without the need for specialized software. Site-to-Site VPN, a private network designed to hide private intranets and all allows users of these secure networks to access each other's resources. Client to server VPN. User is not connected to the internet via his own ISP, but establishing a direct connection through VPN provider. Benefits of using a VPN. Encryption of IP address and protocols. Issues associated with using a VPN. Jurisdiction data leaks. Darknet. Germany has shut down Russia-linked Hydra market, considering as world's biggest and oldest darknet marketplace of illegal items and services. Darknet, also known as dark web, it is that part of the internet which cannot be accessed through traditional search engines like Google, nor is it accessible by normal browsers like Chrome or Safari. It generally uses non-standard communication protocols which make it inaccessible to the internet service providers, okay, or government authorities also. Content on darknet is encrypted and requires a specific browser such as the Onion Ring (Tor). Content on darknet is encrypted and requires a specific browser such as Tor, the Onion Ring, to access those pages. Darknet is itself only a part of the deep web, which includes sites that are protected by passwords part of the internet that is readily available and searchable on standard search engines is called as surface web application, used by journalists and citizens working in oppressive regimes. Researchers and students. Concerns over its use are anonymity, haven for illicit activity, privacy and ethical concerns, drug dealing, communication by terrorists, etc. Deep web, dark web. So surface web, that is Google and Bing, come only 4%. Wikipedia also. Deep web, 90%. For medical records, legal documents, scientific reports, subscription information, competitor websites, organization specific repositories, government resources, financial records, multilingual databases, academic information, 90%. Dark web, only accessible through certain browsers such as Tor. Deep web technologies has zero involvement in the with the dark web. So deep web don't have any technologies involvement in the dark web. Drug trafficking, private communications, political protest, illegal information, TOR encrypted sites. Artificial intelligence. AI is a lot of discussion going on. AI is completely boosting the market. So definition and different types could be asked. Okay. What is AI? Field of computer science that focuses on the creation of intelligent machines that can perform tasks that typically require human intelligence like visual perception, speech recognition, and language translation. Enable computers to learn from experience, adjust to new inputs, and perform human-like tasks. Artificial intelligence is the uppermost part of the whole technology. Any techniques that enable machines to solve a task in a way like humans do is called as artificial intelligence. Machine learning algorithms that allow computers to learn, for example, without being explicitly programmed. Artificial neural networks, ANN, brain inspired machine learning models. Deep learning, a subset of machine learning which uses deep artificial neural networks as models and automatically builds a hierarchy of data representations. Types of AI: artificial narrow intelligence or weak AI, artificial general intelligence, also called as strong human level AI, and artificial super intelligence. Applications of artificial intelligence are automobiles, business, education, finance, manufacturing, gaming, government, healthcare. Generative artificial intelligence. Recently, tech companies around the world are harnessing generative AI for various use cases. Let's see what is generative AI. Describes the algorithms that can be used to create new content like audio, code, images, text simulations. New content could be created. It is called as generative. Achieved by training machine learning models on large amounts of data. Prominent frameworks or models of generative AI like generative adversarial networks, transformer-based models, TBMs, and variational autoencoders. Lambda, language models for dialogue applications, is a machine learning language model created by Google as a chatbot that is supposed to mimic humans in conversation. Like BERT, GPT-3, and other language models, Lambda is built on transformer neural network architecture that Google invented and open source in 2017. It is designed to be able to engage in a free-flowing conversation about virtually endless topics, number of topics. Global Partnership on AI. Okay, launched in 2020, GPAI is an international and multi-stakeholder initiative to guide responsible developments and use of the artificial intelligence. Okay, it headquarters in the Paris, France. 29 member states are the members, and membership is open to all. India is a founding member. Okay, at Organization for Economic and Cooperation Development (OECD) Secretariat it is present. India took over the chair of Global Partnership on AI from France. Responsible AI for Youth 2022. National E-Governance Division of Ministry of Electronics and IT in collaboration with Intel India launched Responsible AI for Youth 2022 program. Program is aimed at enabling school students with artificial intelligence skills in an inclusive manner through exclusive hands-on learning and mentorship opportunities. 4D printing. A renovation of 3D printing wherein special materials is used to print objects and change shape post-production. Need stimuli or triggers to start transformation. Materials used are like hydrogels, thermoresponsive, photo and magnetic responsive, piezoelectric materials, etc. Properties are self-assembly, self-adaptability, self-healing, and shape memory, etc. Selective laser melting, direct link in writing, electron beam melting, etc. Medical, biotechnology, robotics, automobiles, aerospace, textiles, flexible electronics, construction, etc. are the uses. Additive manufacturing or 3D printing. Technology that constructs a three-dimensional object from a digital 3D model or a computer-aided design model by adding material layer by layer. 3D printing is the opposite subtractive traditional manufacturing. It allows creation of lighter, more complex designs that are too difficult or too expensive to build using traditional dies, milling, and machining. Quantum key distribution. Bengaluru-based startup QNU Labs recently innovated advanced secured communication through quantum key distribution systems. About quantum technology. Quantum technology seeks to harness laws of quantum physics, which describes the behavior of matter and energy at the atomic and subatomic level. This is unlike classical physics, which is an object can exist in one place at one time. Quantum computing uses qubits, typically subatomic particles, as its the basic unit of information instead of binary bits. Quantum communication uses quantum bits, typically photons of light for transmitting data along the optical cables, including technologies like quantum key distribution and quantum random number generation. Quantum simulation, quantum sensing and technology. Okay, about quantum key distribution. Quantum key distribution is a secure communication technology that uses quantum physics to construct a cryptographic protocol. It allows two parties to generate a shared secret key. In traditional cryptography, security is usually because an adversary is unable to solve a certain mathematical problem, while in the quantum key distribution, security is achieved through laws of quantum physics. Two such laws are superposition and entanglement. Superposition, ability of the quantum particle to be in multiple states at the same time it is measured. It's superimposition. Entanglement, a situation in which two or more quantum particles are linked in such a way that it is impossible for them to be described independently, but measurements from one particle can be used to instantly draw conclusions about the others. This is entanglement. Sometimes directly questions could come from here. Ethereum blockchain platform switched to merge software mechanism that uses proof of stake mechanisms. Ethereum is a decentralized blockchain platform used to build decentralized apps, DApps, and smart contracts, among others. Merge is a software aimed at slashing energy consumption to the way transactions are validated on Ethereum blockchain. Bitcoin mining term is very important. It is the process of creating new Bitcoin by solving puzzles. It consists of computing systems equipped with specialized chips competing to solve mathematical puzzles. Common centers for mining include China, USA, Russia, and Kazakhstan, countries with cheap electricity rates and colder climates. Proof of work and proof of stake differences. Mechanism Bitcoin uses to regulate the creation of blocks through process of mining. Proof of stake, an alternative consensus mechanism that delegates control of network to the owners of a given token. NFT, that is non-fungible token, is a digital object that can be a drawing, animation, piece of music, or photo or video with a certificate of authenticity created by the blockchain technology. It helps in tokenizing and more efficiently while reducing the probability of fraud. Fungibility refers to an asset's ability to be exchanged with a similar asset without sacrificing its value. Characteristics of NFT are distinct value, digitally read, must be sold or purchased as a whole. Facial recognition systems.